Good evening, students. Today, let us discuss about the topic AIDS. AIDS stands for Acquired Immunodeficiency Syndrome. It is the late stage of HIV infection that occurs when the body's immune system is badly damaged because of the virus. AIDS was first reported in 1981. The disease AIDS is caused by a retrovirus called human immunodeficiency virus. It is a virus whose hereditary material is RNA and not DNA. The virus possesses a very important enzyme which is known as reverse transcriptase enzyme. Now, transcription, it is a process where the information from DNA is transferred into RNA, in messenger RNA. So usually it is a one-way flow of information, that is from DNA to RNA. But this virus possesses an enzyme which is known as reverse transcriptase. These enzymes have the capability of reversing this process. That is, from RNA, the information is transcribed into DNA. That is, the flow is reversible due to the action of this particular enzyme, that is reverse transcriptase. So here we can see the diagram which shows the structure of HIV. It has a protective glycoprotein coat. The nucleus, it does not comprise of a nucleus, but you can see that it is just suspended. And this is the RNA and this is the reverse transcriptase enzyme. Now let us discuss the mechanism of infection by HIV, or we can say life cycle of HIV. Here we have a diagram which is showing the life cycle of HIV. Any virus, when outside the body, acts as a non-living organism. But when it enters into the body and when comes in contact with cells, it becomes a living organism and starts its life cycle and infects the body. So this is the HIV. The HIV, when comes in contact with a cell, this is not any cell, but these cells are those which protect our body, from any microorganisms. That is right, the T cells or the macrophages. So when HIV comes in contact with a T cell or a macrophage, it releases the RNA into the cell, as you can see here. This is the cell, this is the virus. It is releasing the virus into the cell. Also, along with the viral DNA, the reverse transcriptase enzyme also enters the cell. Okay, so viral RNA is introduced into the cell, and also the reverse transcriptase. Now, in the presence of reverse transcriptase enzyme inside the host cell, the RNA is transcribed to produce DNA. Then this viral DNA is incorporated into the host's genome, as you can see here. Then new viral DNA is produced by the infected cells. That is known as DNA replication. Next, the viral DNA which is incorporated into the host genome also undergo transcription along with the host genome, thereby producing proteins, viral proteins. These viral proteins now incorporate to produce new viruses inside the cell. New viruses which are produced are released from the cell and attack new cell. In this way, the virus infects a cell. The virus, when infects a cell, that cell dies. Okay? So in this way, the life cycle of HIV takes place. Next, we discuss about the transmission of HIV. First, sexual contact with infected person. So, a person should avoid sexual intercourse with an infected person. Next, when blood is given or blood transfusion take place, the blood should be checked whether the blood is infected or not. Sharing infected needles, razors, etc. from an infected mother to fetus through placenta. Now, how can you guess, or how can you understand that a person is being affected? The signs and symptoms: swollen lymph nodes and fever, decreased count of blood platelets, sweating at night and tiredness, weight loss and loss of appetite. HIV may attack nervous system, causing damage to brain. It may lead to memory loss, loss of coordination, partial paralysis, mental disorder, and loss of ability to speak and think. The person becomes susceptible to other diseases. Next, what steps can we take to prevent these diseases? Use of condoms during sexual intercourse. During any blood transfusion, the blood to be transferred should be tested. Use of disposable syringe, razors, etc. Controlling drug abuse. Advocating safe sex and promoting regular checkup for HIV. Now, the most widely used diagnostic test for AIDS is ELISA, that is enzyme-linked immunosorbent assay. There is another method which is known as PCR, that is polymerase chain reaction. Thank you.